Moving on in chapter 15, this is formally the part 2 because it's the second PowerPoint that I created for this chapter. As usual, I break these things up over labs and lectures, and so the same story is sort of here. As always, if you have any questions, you're having any trouble, please let me know and I'll be glad to help you. One thing to note is that with these lectures and this presentation is I'm always trying to review stuff. So there are some slides today that are sort of bringing some of the things we've already talked about back into the picture. So this lecture, or perhaps 2 lectures, will be covering these 4 elements of the chapter today. Regulating cardiovascular function, that is to say, how do we change cardiac output and vasoconstriction dilation? This is basically a rehash of chapter 11, sympathetic, parasympathetic. We are going to talk about capillary exchange just a little bit, but this is more of an intro into what we're going to be doing in the next unit. Just a little bit about the lymphatic system, not a whole lot. And then just one of the issues at the end is about edema. I could talk about cardiovascular disease, and there's certainly a lot to talk about there, but I've elected to skip that and instead talk about another topic, which hopefully will make some sense to you given that we're talking about exchange at the capillaries. We talked about this slide last time. Remember that we are dealing with factors that influence pressure. First of all, blood volume, how much fluid intake versus how much loss, and how we can take in and lose. Intake is almost exclusively gastrointestinal, whereas loss can be passive because of evaporation or fluid loss for certain reasons, or it can be regulated through the renal excretory pathway. The effectiveness of the heart, and this is fundamentally cardiac output. The heart rate times how much blood it squeezes into the cardiovascular system every time it squeezes. Resistance, the diameter primarily of the downstream arterioles, meta-arterioles, and precapillary sphincters. Not a lot of resistance is attributed to the aorta or some of the large arteries. Those dilations and constrictions are generally for sustaining blood pressure. That is to say, the pressure reservoir term that we talked about in earlier sections. And again, the radiuses to the 4th power is the big one here. That's the one we talk about, those arterioles, meta-arterioles, and precapillary sphincters. Now, there is this thing called myogenic autoregulation. I'm going to save that term. It's something you will have to know in Unit 4. Basically, it's the relaxation during the systolic wave followed by constriction. That's another term for the change in vascular smooth muscle tone during different phases of systole and diastole. But we'll talk about that in particular, we'll talk about in the kidney. So it's a term you want to keep on your radar, but it's not until next unit. Lastly, how much blood is returned to the heart by virtue of the smooth muscle tone in the large veins. If they increase in their tension, they will send more blood back to the heart. That would increased venous return. But we can also see this by skeletal muscle contraction, which squeezes veins, reducing their diameter, again, sending more blood. We call that collectively venous return. And I'm reinforcing another figure that we talked about previously, just to kind of, again, to bring back the mechanisms that you've already learned about in this class. On the fast response side, if we see a change in blood volume, in this case we're saying an increase, but we could see the opposite when it comes to a decrease. This is driven by sympathetic and parasympathetic signaling. Either changes in cardiac output, increased strength of contraction, increased rate by sympathetic, or decreased strength of decreased rate. Really, with parasympathetic, it's almost entirely rate, not so much strength. Vasoconstriction, vasodilation follows the same story. We dilate to lower blood pressure, and we constrict in order to raise blood pressure. That's true of the arteries and of the veins. Then we have the slow side of the equation. This is more cytokine and hormone driven. This is driven by the renal excretion pathway, limiting or increasing fluid loss. Both situations are negative feedback loops. That is to say, they take us back to a basal set point, back to a homeostatic baseline, and we resume normal function. So let's look at the rapid response pathway, since we've already done reflexes like this in the past in chapter 8 with neuronal control, with sensory responses, sympathetic and parasympathetic. So this is just an extension of something we've already done. I'm just going to show you now how that applies to blood pressure, cardiovascular function. So we're going to look at a change in blood pressure, how it responds in the receptors, how it's integrated, and then we have an effector. So the initial stimulus is a change in blood pressure, and in this figure we could have a change up or we could have a change down. It's not going to matter because we're going to show you both situations. The receptors in this pathway are mechanoreceptors. That is to say, they get stretched when blood pressure is higher, or they retract and are less stretched when pressure is lower. And we call these baroreceptors because they are sensitive. Their adequate stimulus is mean arterial pressure in the inside the vessels. Baroreceptors, mechanoreceptors. And here's an old anatomical image showing you right where the carotid baroreceptors are located. Okay. The other one is found in the other baroreceptors are found in the aortic arch. The more they are stretched, the more action potentials they send to the central nervous system, and the less they're stretched, the fewer action potentials are sent. And so here we are back into our original figure. So we have a change in blood pressure, which causes a change in the action potential pattern. Okay, so this is sort of it's a sort of a frequency coding, if you will, frequency coding. How many action potentials go to the central nervous system? In this case, they are sent to the medulla, where there's an integration center that interprets the number of action potentials and then chooses the appropriate response. This medullary cardiovascular control center is known as the MCCC. And again, it interprets the frequency coding of action potentials from either the carotid or the aortic baroreceptor. If we have lots and lots of action potentials being sent here, the Medullary Cardiovascular Control Center, MCCC, will then trigger the parasympathetic pathway in order to lower the mean arterial pressure. So it's going to send signals out, these effector neurons in the parasympathetic pathway. So the parasympathetic, we also have a sympathetic side. I'll get to that in just a second. So now let's see the parasympathetic side. The parasympathetic side primarily triggers the sinoatrial node, the SA node. So they're going to activate the SA node, and in doing so, they affect the funny current. They affect I sub F. What do? They slow the kinetics of sodium inflow into those autorhythmic myocardial cells. So we're going to slow the kinetics. The slope, instead of being so steep, is now flatter, takes longer to reach action potential. Now, indirectly, more through hormonal regulation, we can also affect the constriction and dilation of arteries. So we can get a little bit of calcium regulation through parasympathetic activation to dilate those guys. We might see some dilation in the veins in order to reduce venous return, and so we get reduced blood pressure. So what if we have a drop in blood pressure that causes fewer action potentials to be sent to the MCCC? In that case, we're going to want to increase response to that in order to bring blood pressure back to that homeostatic set point. In this case, it will be the sympathetic side. Even if we're relatively at rest, if our blood pressure is too low, we're going to get a little sympathetic kick in, and that's going to bring our blood pressure back to where it should be. The sympathetic, first of all, activates through the sinoatrial node, the SA node, okay, steepening that I sub F, steepening that funny current. So more sodium comes in quicker, and that leads to more frequent action potentials from the SA node and a faster heartbeat. The sympathetics can also, unlike the parasympathetic, can affect the ventricles as well and modulate strength of contraction. And that is driven by calcium kinetics. So if we let more calcium in the myocardial tissue, you will get a stronger contraction. So we can regulate. We don't recruit muscle fibers like we do in skeletal muscle. We vary strength of contraction in the heart by either regulating calcium kinetics or by the length tension relationship we call the Frank-Starling law. Okay. On the skeletal side, and I know I'm going a little bit of an aside here, we vary strength of contraction by recruitment and also the length tension relationship. We don't selectively recruit myocardial fibers in the heart. We regulate it by either how much calcium we let in, the calcium kinetics, or how much stretch there is. Now, this isn't the end of the story with the sympathetic. We can also affect arterial smooth muscle tension as well as venous smooth muscle retention with calcium regulation as well. By increasing the calcium, we increase the contraction in the smooth muscle, and that's true for the arterioles, but it's also true for the veins. So we have more venous return, we have more internal resistance coupled with cardiac output, blood pressure will rise. Take it slow, work through this, start here, work here, work here, and then follow through. Being mindful through here, that mindful intention of understanding what's going on is what's going to get you through figures like this, not only now, but down the line when you're dealing with therapeutics and clinical outcomes. And this is just another slide just kind of explaining that. The sympathetic side, that's the accelerator nerve. I'm not going to test you on that. Affects the SA node. We have other nerves that affect calcium kinetics, but what you don't see here is a vagus, another parasympathetic going to the ventricles. Parasympathetic is SA node only. Sympathetic is SA node as well as neurons that will affect calcium kinetics in the myocardium. But the key signal for our purposes is funny current, I sub F channels. That's our mechanism for cardiac output this time around. So a little bit of review here, Again, playing out some of the scenarios. Cardiac output is the heart rate times the volume that's injected every time the heart squeezes. We also have tension and relaxation here in the arteries, which sustains pressure, not involved with resistance. That's the pressure side of the equation. The resistance side of the equation is in the arterioles, meta-arterioles, and precapillary sphincters, total peripheral resistance. So if we have increased peripheral resistance and we keep cardiac output the same, that would mean an increased mean arterial pressure. If we have an increased mean arterial pressure, the baroreceptors are going to fire more often, send it to the MCCC. That's going to fire off what we call the baroreceptor reflex, in this case a parasympathetic response. That parasympathetic response is going to slow the heart down, decreasing cardiac output. We will see some ancillary effect in the peripheral resistance, but mostly when it comes to the parasympathetic, it's the cardiac output. And so by reducing cardiac output, we return mean arterial pressure to normal. Now let's move to the capillaries, what we call exchange epithelia. They are facilitating gas and nutrient exchange between tissues and the bloodstream. So they're very, very thin. Again, simple squamous epithelia, very, very thin to facilitate diffusion, transcellular transport, and especially paracellular transport when it comes to most normal capillaries that we find in tissues. Turns out that tissue that is very heavily metabolic, like for instance liver tissue, brain tissue, skeletal muscle, tends to have very, very high capillary density, whereas places like adipose tissue and other tissues that are less metabolically active, you see a much lower capillary density. And this can change if the metabolic activity of the cells change. So if you are working out more and your myocardium's working more, you're going to see an increased capillary density. Same is true in your skeletal muscles as well. Capillaries have those thinnest walls. Why? Because diffusion is limited by distance. Even paracellular movement is distance dependent because it is a function of diffusion in some ways. So it's a single layer, very, very delicate. There is a basal lamina on the outside of those capillaries that helps let the endothelial cells know that they're in the right place and they shouldn't grow or divide. We have two, and actually three, major categories of capillaries. We have what are called continuous capillaries, which I'm going to talk about in a second. We have fenestrated capillaries, and then we have these funny ones called sinusoidal capillaries, which I'm not going to talk about. Book doesn't talk about it. I'm not interested. So we're going to talk about continuous, and we're going to talk about fenestrated. Continuous capillaries are like pavement stones, but they are leaky. That is to say, these junctions in between the capillaries have relatively, quote-unquote, permissive tight junctions. They let lots and lots of material pass paracellularly. Yes, you do get transcytosis. You may even create these sort of transient channels, if you will. But the vast majority of material is actually moving paracellularly between the cells and not through the cells. You see both processes, and they're important. Nevertheless, you do see mostly paracellular movement in continuous. And this is by far the largest population of capillaries in the body, far and away. Fenestrated capillaries are what we're going to talk about when we get to the glomerular capillaries in kidney. Fenestrated, there are very few of these, mostly the glomeruli in the nephrons. But fenestrated have these special pores. They are called fenestrations. They are structures in the cells that facilitate paracellular movement, facilitate what we call bulk flow. Bulk flow meaning that we're going to move things by virtue of pressure gradients rather than pure osmosis. So we see fenestrated capillaries when we want to filter things. And then, like I said, the glomerulus is a big example of it. Now, that's to say there is still transcytosis going on, and you still see these transient fenestrations or pores opening. But by and large, we're talking about paracellular bulk flow type movements in fenestrated capillaries. And again, the number one place, the glomerulus of the kidney. I've often thought about putting this figure earlier on in the chapter, showing you the velocity that we see in blood vessels versus capillaries and so forth. What we see is a very, very high velocity in the aorta. Even though it has a large cross-sectional diameter relative to a capillary, there's only one aorta. So the total cross-sectional area is only about 1 square centimeter. And so the velocity is very, very high. As we bifurcate, the total cross-sectional area begins to rise, and we bifurcate more, the number rises more and more. So we're seeing a larger sum cross-sectional area when we get to arterioles, meta-arterioles, and especially capillaries. By the time we get to capillaries, we're at 5 square meters, which is a huge surface area. Okay, that's roughly the size of a tennis court on one side. And consequently, velocity is very, very slow. This facilitates exchange. You don't get a lot of exchange in the arteries or the aorta. Hardly any at all. Why? Because the blood's moving so fast, you don't really have time for exchange. Yeah, there's going to be a little bit. There's always going to be a little bit going on, but not a lot. Where we see the real exchange going on is right here in the capillaries. That's where we have by far the largest cross-sectional area and by far the slowest velocity. As we condense the vessels, again, cross-sectional area begins to diminish and velocity begins to increase again. So the velocity is a function of the sum of the cross-sectional area of the vessels. The more vessels you have, the more cross-sectional area you're going to have, and the velocity is going to subsequently be slower. A reminder slide about the idea of total cross-sectional area, and that velocity is not the same as flow. So when we're talking about capillary exchange, what we're talking about is movement between two different compartments. The fluid inside the blood vessels we call plasma, and the extracellular fluid outside the blood vessels known as interstitial fluid. There are important differences between these two. We see a lot more protein in blood plasma than we do in interstitial fluid, so there are some compartmental differences. We see a lot of paracellular movement when it comes to capillary exchange, but occasionally we will see endothelial transport as well, but that tends to be very situational and specific. So that transcellular transport is what we would refer to as endothelial. It's moving through the cells. See a lot of that in the blood-brain barrier. When we're talking about small molecules, they diffuse the most easily. Or if they're very lipid soluble, they'll diffuse the most easily. So oxygen, carbon dioxide can readily move through paracellular or transcellular. They depend on concentration gradient, which is why blood flow, Q, is so important, because Q, blood flow, perfusion, sustains gradients so that if we have nutrients that we want to move into a cell, they're moving in a relatively unidirectional way. We don't want to achieve equilibrium when it comes to capillary exchange. And there are some anatomical and biophysical ways of manipulating capillary exchange that I'm not really going to talk about this semester. Cool stuff, though. Larger molecules, proteins, if we're going to need to move them, are going to move through transcytosis, transcellular, endothelial transport. They're going to be transcellular in nature. And that's part of the reason why we don't see as much protein in the interstitial fluid, is because you can't move them paracellularly. They have to be moved by transcellular processes. And again, paracellular is between cells. Between cells. In the case of transcellular, that's moving through cells. And again, we have arrows going from left to right. If we're talking about waste products or things out here, it would be from right to left. And so exchange occurs in both directions. Okay? In both directions. It just depends on what we're talking about. So with capillary exchange, that is to say the movement between blood plasma and the interstitial fluid, whether it's from the blood plasma or from the interstitial fluid, utilizes certain mechanisms that we have talked about in the past, but I'd like to refresh a little bit in order to set the stage for the dynamics of capillary exchange. So we have bulk flow, and that's the mass movement as a result of hydrostatic pressure. Yes, osmotic pressure can play a role in this as well. So if you have differential osmotic pressure, you can get bulk flow. But most of what's going on with bulk flow in this situation is hydrostatic. That is to say, the ambient pressure between the blood plasma and the interstitial fluid. But it's pressure gradients that drive this. It's not concentration gradients. Filtration utilizes hydrostatic pressure. Okay, so it's a bulk flow process. Filtration is fluid movement out of the capillaries into the interstitial fluid. And so hydrostatic pressure drives that. Mean arterial pressure drives filtration. That is to say, we are pushing things through those paracellular spaces. It is filtering things. Proteins generally do not get through those paracellular junctions. So we are filtering, just like a coffee filter doesn't allow the grounds to filter through on its way to the cup of coffee. So it's hydrostatic pressure that drives filtration. And that usually works towards what we call the arterial end. And I'm going to show you here in a picture in just a second what I mean. So hang on to that. We see more filtration than the next mechanism. The next mechanism is absorption, and that's movement from the interstitial fluid back into the capillaries. So we're going from the tissues back into the capillaries, and this is known as absorption. This is driven by osmotic pressure gradients, okay, as opposed to hydrostatic. It actually opposes hydrostatic pressure. Where we see net absorption is closer to the venous end of a capillary, where the pressure is a little bit lower. And again, that's osmotic pressure of bulk flow, pulling fluid back from the tissues back into the capillaries. So let's take a look at the picture, and this hopefully will begin to make sense. So here's our arterial, meta-arterial, precapillary sphincter, and so forth. Right here versus over on the right side of this picture, we have a higher pressure. We have to have a pressure gradient or the blood's not going to move through here. So there's more ambient hydrostatic pressure at this end of the microvascular circuit. And there is less pressure over here on the right and the venous side of the microvascular circuit. In the beginning, we have relatively high mean arterial pressure. That means that the majority of the dynamics between tissue and the capillaries is filtration. That is to say, the majority is moving out of the capillaries into the tissue. Things like water, things like nutrients and so forth are moving by primarily paracellular movement through the junctions between the cells into the tissues. As we move further, the fluid level is going to drop, and so the pressure is going to drop. More resistance occurs, pressure is going to drop. And when we get over onto the venous side of the microvascular circuit, now there is more osmotic pressure pulling fluid back in than hydrostatic pressure pushing out. And so we have net absorption on the venous side of the capillaries, whereas we have net filtration on the arterial side. Now, there is an imbalance here that usually during the course of a 24-hour period, we're going to have more stuff move out of the capillaries than move back in, to the tune of about 3 liters per day. There is 3 liters more fluid that moves out of the capillaries than moves back in, and that leads to an imbalance. And so we have structures known as lymphatic vessels. They're specialized vascular structures, very similar to capillaries in many ways, very similar to veins when they get larger. But one of their roles is to capture that excess fluid and return it back to the bloodstream. So lymphatics, one of their roles is to return excess fluid from the tissue back into the bloodstream. But there's a net of about 3 liters per day of filtration versus absorption. And so what are the forces at play here? And this can look a little bit complicated at first. I'm going to work very, very slowly here in order for you to understand the different abbreviations and to understand this. We're going to see this figure again when we get to kidney, and it's going to actually have one more force involved. Okay. In the previous slide, we said we had relatively high pressure on this side. P sub H is hydrostatic pressure, otherwise known as mean arterial pressure. Okay. Mean arterial pressure, hydrostatic pressure. That's the force that of the plasma pushing against the walls of the capillary, and it's relatively high. The Greek letter pi, and that's what that is, the Greek letter pi, you can see it here and here and here and here. That Greek letter pi is colloid osmotic pressure. That is the osmotic pressure that wants to pull fluid out of the tissues back into the capillary. That would be the absorptive force. Okay, so P sub H is the filtration force. Pi is the absorption force. And so on the arterial side of the microvascular circuit, we have 32 millimeters of P sub H in this example. So we have 32 millimeters of mercury pushing out of the capillaries, and we have 25 millimeters of mercury pulling in. That's the colloid osmotic pressure. That's the problem. And so 32 is higher than 25 by 7, and so we're going to have net filtration of fluid out of the capillaries because P sub H is higher than pi. As P sub H drops, as we get further down the microvascular circuit because of friction, because of loss of fluid, P sub H and pi will become equal, in this case 25. And so there is no net exchange in the middle of that microvascular circuit. And then finally, when we get to the venous side, P sub H has lost a significant amount of pressure, so it's at 15. You can see the arrow is smaller than over here on the left. So P sub H out is 15, but pi is still 25. It still wants to pull fluid in. And so we have net absorption going on. We have net absorption, so we're pulling fluid back in. Even so, even though we have a 10 over here versus a minus 10 and a plus 7, there's still a net excess of filtration versus absorption to the tune of 3 liters per day, and that's collected by the lymphatics. So if you can look at this right here, this figure question brings up some fun stories for me that I'll tell you about here in a little bit. Okay, so read this question. Suppose that the hydrostatic pressure at the arterial end increases from 32 to 35. If P sub H remains 15 at the venous end, how does that change or affect filtration? Let's talk about that in the next slide. A practical example of the answer to the question in that previous slide is shown right here, which on that wonderful day, if you decide to get married and you find the right person and you take your wedding vows, or perhaps you might notice it when you're nervous or stressed out about something, like this young woman who's doing her best to catch the coronavirus. Or even after an extended period of working out, if you've been at elevated cardiac output for a period of time, like for a long run or something like that, you might notice that it's difficult to put rings or take rings off of your fingers. And that's absolutely the case when you get married. On the day you're really, really nervous, you're in front of your family and friends, and it's time to put the ring on, and it doesn't go on. Okay, don't freak out. It's actually quite normal. Anytime you have an elevated blood pressure because of anxiety or excitement, good or bad, that's going to cause increased filtration out of the capillaries. That's going to lead to something known as edema. And so we have edema anytime that we find ourselves under sustained mean increases in mean arterial pressure. The fluid out of the capillaries is going to exceed even more so than the fluid back in. So one of the mechanisms of the lymphatic system is to help handle that excess fluid coming out of our capillaries, either the normal amount of fluid or even more fluid than normally is. But during those transient times of stress or elevated pressure, you will see transient edema. So mechanism, or shall I say, function number one, is to return fluid and or proteins. Occasionally you break down proteins or proteins spill out of the capillaries. Return those back to the circulatory system. Role number two of the lymphatic system is actually what we find in the gastrointestinal tract. You don't really see it in this picture. All you're seeing here is a sort of the nodal and the vessel lymphatics, but there's a whole network of lymphatics called lacteals, which are found in the gastrointestinal tract, and those lacteals are specialized in order to pick up fat droplets. And so they use penocytosis in order to bring up fat droplets out of the gastrointestinal tract and to bring it to the circulatory system, where they can then be used by the liver and or other tissues. The third mechanism, or the third function of the lymphatic system is primarily in these nodes or in the spleen or in the thymus, and that is an immunological function. We say it's a filter for pathogens. It's also a quote-unquote filter for cancer cells. We say filter because what happens is when material moves through these nodes, they come into close contact with numerous numbers of white blood cells, what we call lymphocytes, or in some cases thymocytes. Those cells are there to surveil the material and to spot and respond to pathogens. And so lymph nodes serve as active areas of immunologic function. But we're going to abbreviate that as a filter, if you will. So return fluids and proteins to the circulatory system, pick up absorbed fat that we absorb from the gastrointestinal tract and bring it to the liver and then on to the rest of the body. And then lastly, immunologic function, serving as a quote-unquote filter for pathogens. And so let's bring some clinical aspect to what we've been talking about. And I've been alluding to it, or I've even mentioned it in the last 3 or 4 slides. But let's talk about edema because it is a major clinical sign of disorder or disease, inflammation. The major causes are: one, inadequate drainage of the lymphatics, either because the lymphatics are occluded or the lymphatics are malformed or for some other reason they can't drain. And so we have inadequate drainage. And so the fluid is going to filter out of the capillaries faster than it is resorbed back into the capillaries, and so excess fluid remains outside of in the interstitial fluid than ought to be. The other major way is that filtration far exceeds absorption. And this gets back to my wedding ring analogy or my workout analogy or my stressed out analogy. In periods when we have elevated hydrostatic pressure, that will lead to faster filtration than absorption, and thus we will get fluid accumulation. Two other mechanisms in this filtration are if we have an osmotic issue. If you have liver disease and you can't make the osmotic proteins that drive osmotic pressure, then you can't pull fluid back into the capillaries. If there's no osmotic gradient, then there is no pi in that force. Okay? The third possibility is that if you have, for whatever reason, an increase in proteins outside the blood plasma, that can be due to hemorrhage. That can also be due to breakdown of proteins outside the plasma that lead to a change in the osmotic pressure. So these two right here are about pi, and this first one right here is about P sub H. But let me explain what we mean by an increase in interstitial proteins, and I'm going to use this figure over here in the lower right corner. This child suffers from a disease known as kwashiorkor. Kwashiorkor occurs generally in young children who have recently stopped breastfeeding. What's happened is their body has switched from a caloric intake that's relatively balanced between carbohydrates and proteins and fats to almost exclusively carbohydrates, and usually insufficient carbohydrates at that. They are weaned from their mothers, they can no longer breastfeed, and so their calories change dramatically, and they're exclusively carbohydrates and probably at a subsistence level. Notice his belly is distended. Even though you can see general signs of malnourishment through his body, his belly is distended almost as if it's full of fluid. What's going on is the cells are breaking down connective tissue proteins. They're breaking down collagen, they're breaking down fat, they're breaking down unnecessary, quote unquote, proteins to use for energy supply. When you break down proteins, you create protein fragments, which are osmotically active. You create you have amino acid byproducts, which are osmotically active. And so the osmotic balance between his blood vessels and the extracellular fluid in his belly is such that the fluid is filtering, but it is not, but pi has been interfered with, and so the fluid is not going back into the capillaries, exceeding the ability of the lymphatics to return that. Okay? That's what's going on in this case with kwashiorkor. So this concludes chapter 15. So we have one more chapter to go in this section, and that's on blood. Thankfully, though, that's a relatively short chapter. We've had some big chapters, 12 and 14, and now 15, and one more to go for this unit. Thank you for your time, and I will talk to you again soon. As always, if you have questions, reach out to me, let me know. I'll be glad and thrilled to be able to help you out. Thanks.